Good evening. My name is Candace and I'm an event manager. On behalf of Town Hall Seattle and our friends over at the Institute for Systems Biology, it's my pleasure to welcome you to tonight's live stream conversation with microbiome researcher and professor Dr. Eric Alm in conversation with Dr. Sean Gibbons. As we get underway, I would like to acknowledge that our institution stands on the unceded traditional territory of the Coast Salish people, particularly the Duwamish. We thank them for our continuing use of the natural resources of their ancestral homeland. And thank you all for tuning in. Town Hall is proud to be a community-focused organization, a place where we can share and sustain ideas and creativity, even if we can't gather in person. I'd like to thank Dr. Alm for appearing tonight to help make that possible. Town Hall will continue to produce virtual content this fall, including our podcast, In the Moment, which features exclusive guests, and many of our past talks are available in video or podcast form in our digital media library. Town Hall is adding new events and podcasts every day. Upcoming programs include Sean B. Carroll and the Improbability of Existence, Yolande Stangers and Jenny Kennedy with the proposal for changing our feminized digital assistants, and Jane Fonda and Elizabeth Lesser about the superpower of women, and many more. Town Hall and the nonprofit community at large have been put under significant strain due to the ever-changing landscape. We hope you'll consider extending your generosity to help support us during this difficult time by making a donation or becoming a member. You can click on the donate button at the bottom of your screen at any point during the program. For viewers who want to watch this broadcast with closed captioning, we recommend viewing the stream via our YouTube page. It's linked here in the chat. To enable real-time closed captioning, click the CC button in the bottom right corner of the video player. The video will be available for rewatching immediately following tonight's broadcast, both on YouTube as well as on Crowdcast here. Tonight's presentation will be about 60 minutes, including Q&A. Our speakers will select questions from those submitted in the Ask a Question field and also from the chat on our YouTube page. We can't guarantee that we'll be able to address every question, but we will try to get to as many as possible. Please keep your questions concise and in the form of a question. Town Hall's work is made possible through your support and the support of our sponsors. Our Arno Matulski Science Series is supported by Microsoft, KUOW, Wyncote Foundation Northwest, and the taxpayers of Washington. And finally, Town Hall is a member-supported organization, so I'd like to thank the members watching tonight. We're so excited to be working with Institutes for Systems Biology this season. I'd like to introduce Dr. Sean Gibbons, Assistant Professor and Washington Research Foundation Distinguished Investigator at ISB. He completed his postdoctoral training with Dr. Alm's laboratory in the Department of Biological Engineering at MIT. So please join me in welcoming Sean.
Thanks, Candace. Good evening, everybody. Welcome. Like Candace said, I'm Sean Gibbons. I'm faculty at the Institute for Systems Biology. ISB is a nonprofit biomedical research institute located in Seattle, and we're an affiliate of the Providence Health System. ISB focuses on some of the most pressing issues in human health, including COVID-19, brain health, cancer, sepsis, and aging, as well as many other chronic and infectious diseases. ISB also focuses on improving K-12 STEM education and, in particular, addressing inclusion and equity for students of color, women, and other historically underrepresented groups in science. You can learn more about ISB by visiting our website at isbscience, all one word, .org. Before we begin, I'd like to express our thanks to our partner, Town Hall Seattle, for several events that we'll be doing over the course of this fall, and to the sponsors who helped make this event possible, Metabolon and the Washington Research Foundation. Tonight, I'm super excited to welcome Dr. Eric Alm. Dr. Alm is a professor of biological engineering at MIT and the Broad Institute and co-director of the MIT Center for Microbiome Informatics and Therapeutics. His lab develops complementary computational and experimental methods to engineer the human microbiome. As a side note, like was mentioned earlier, I was fortunate to do my postdoc work with Eric a couple of years ago. Dr. Alm's work over the past several years is focused on the human microbiome, and his group has pioneered approaches to standardizing fecal transplants for the treatment of C. diff infections, and developing a wider range of so-called microbial therapeutics or pharmaceutical grade probiotics. Recently, his lab has moved into analyzing sewage to track population health. He is the co-founder and scientific director of Biobot Analytics, a company that transforms sewers into public health observatories. Biobot recently launched a wastewater epidemiology program to map COVID-19 outbreaks at the population scale, developing efficient tools and techniques to monitor SARS-CoV-2 in sewage. Initial results suggest that these data can be used to help guide public health officials and track and manage the pandemic. Following Dr. Alm's presentation, he will join me for a Q&A with about a number of topics related to COVID surveillance and detection, in addition to questions that I will be asking. If you all have questions at home, please type them in under Ask a Question on the Crowdcast, and we'll get to your questions as well, hopefully. So, sorry for all that spiel. Without further ado, I'm honored to welcome Dr. Eric Alm. So Eric, the floor is yours.
Great. Well, thanks so much for that introduction, Sean. It's really, it's a pleasure for me to be here. I love Seattle. I actually did my PhD with David Baker at the University of Washington. And so Seattle always kind of holds a special place in my heart. I want to tell you a little bit about, before we get kind of right into the COVID surveillance, I want to tell you a little bit about how we got here and how I got into this business of sewage surveillance. So it started a few years ago when I was having conversations with the Sensible Cities Laboratory here at MIT. And they were interested in some of the work we were doing with the microbiome. And so, as Sean mentioned, the microbiome, that's all of the biological organisms, really the ecosystem of microorganisms that live in and on our body. And they had the idea, because they study cities, what about the microorganisms that live in and on cities? And so we started to think about what would be the possibilities. What if we could monitor all of the microorganisms? And then we thought, we don't really even have to stop there. In addition to looking at the biology, we can look at the chemistry. And when you think about it, the largest collection of mass moving in and out of a city every day, you might think it's, planes or cars or freighter trucks, or maybe if it's a port city, maybe it's boats, but it's not. It's water. There's more water moving in and out of a city than really anything else, any sort of like man-made transportation system. And so generally it's pure, clean water, mostly used for drinking and showering and washing our clothes and other industrial uses, that's being piped in. And then, as a society, humans do things to that water, and almost all human activities leave some residue in that water. And I started to think, why aren't we mining that water to get some better insight into, what all the human activities are that are going on in the city? And that was really kind of the birth of this academic project called Underworlds. So we can think about what would we do with it? Well, one really important application, really our main focus was public health. Public health epidemiology, it's really hard work, and part of the problem is there are all these different people and all of these different problems that we're trying to solve. And just to study one question, one public health question, we need to recruit a cohort of individuals. We have to be careful to make sure that cohort is representative of the society at large. We have to convince those folks to give us samples, especially if we're talking about like urine samples or stool samples. Sometimes people are squeamish about that. I'm always surprised that people are more squeamish about donating a stool sample than a blood sample. To me, the blood sample seems much more invasive. But folks are squeamish about it, and then we also have issues related to, the keeping that healthcare data private. And so all of these things kind of come together, and they make it very difficult to do public health studies. They make it not only expensive, but it requires a lot of hands-on time by the folks collecting the data and processing it. And it takes time to recruit patients and or subjects in these research studies, get them up to speed, get their consent to use their data, explain kind of what things are for. And then when we collect the data, it's not we have to make sure it's anonymized and it's kept private. And so we started to think, well, all of this data is really it's in the sewers, right? It's in the wastewater. So this is a picture of the Deer Island wastewater treatment plant. So this plant serves about 2.3 million people that live kind of in and around the Boston area. It's the second largest treatment plant in the United States. And I think it receives about 1.3 billion liters of, on average, water every day. And that water, like I said, carries some traces of all the human activities that are going on kind of in and around the Boston area. So what if we could take that wastewater and then translate the biology and the chemistry that we find in that wastewater and use that to get an estimate of public health? So here's a classic picture. This is an image that was, drawn by hand by Dr. John Snow to track the Vibrio cholera outbreak in London. And what he did was he took all of the cases of folks that came down with cholera, and then he put little black dots on where they lived. And after making this map, what you can see about this particular region of London is that the cases seem to aggregate around the street in the middle. And if you zoom in there, it'll say Broad Street, okay? And there was a water pumping station where folks would get clean water on Broad Street. And so what he was able to deduce from that is that maybe the Vibrio cholera was living in the clean water supply. And so they shut down the pump. And that coincided with a decline in the cases of cholera in that region of London. And we said, this is really exciting. It was an amazing example. It was in the mid 1800s, but this is basically how we're doing the same thing today, right? We haven't really advanced, and I think that the sewers offer us an opportunity to really take this basic idea, but modernize it and bring it into the digital era. So we could imagine collecting wastewater from different manholes all around, let's say, so here's the city of Boston, and then we could look at the wastewater in all those different locations and see which locations correspond to different factors that we want to study from a public health perspective. So that might be an infectious disease. It might be pharmaceuticals. It might be illicit drugs. It might be what genes are carried by bacteria. Are there bacteria that harbor antimicrobial resistance genes that are, slowly making our antibiotics less and less effective? Could we use the sewers to kind of monitor all of these things and give us a sense of basically an early warning, an early detection system? So we thought of a number of different uses. One might be, kind of doing epidemiology, trying to figure out maybe where outbreaks are happening. We might use it for real-time outbreak mitigation. And I think, we'll probably have time to talk about how we're just starting to implement that now in light of COVID. We can use it to just generally better inform public health. Our students and generally people in different neighborhoods, are they getting the nutrition they need? Are there just general health biomarkers that are really elevated in some parts of the city, but really depressed in others? And can we find correlates that might explain that? And can that help us identify, if we have limited resources, where we should intervene and try to improve nutrition and improve health generally? And ultimately, we'd like to have some kind of early warning for the next biological threat or chemical threat after COVID, right? So has this been done before? It has, and I think really the exemplar of this approach was Israel in 2013. So in 2013, they were doing polio surveillance in wastewater in Israel, and they started to identify for the first time in that project positive cases of polio in the wastewater. Now, polio has basically three properties that make it pretty ideal for wastewater-based surveillance. And not every human pathogen is necessarily a good target for wastewater surveillance. But polio kind of has all three. So the first is poliovirus is shed in pretty reasonable amounts in wastewater, so in either urine or feces. Secondly, there's a large asymptomatic population. So for every one kid who ends up getting paralyzed because of polio, there's 99 that are basically asymptomatic. So that means that we have a good shot of seeing the early warning, seeing those 99 kids, and maybe stopping it before any kid ends up with paralysis. And then the third box that we like to check to make sure it's a good wastewater-based epidemiology target is that information actionable in any way? Okay, so it's fine if we can say, hey, look, we think there's an outbreak. The cases are increasing, but if knowing that in advance doesn't, really help us do anything to prevent it, then it's interesting, but it's not a great target. It's not useful. In the case of polio and in Israel, they were able to switch to the live attenuated vaccine, the oral vaccine, which is better at stopping transmission. And they basically immunized, I think, something on the order of 1.4 million kids within a few months of finding that in the sewage. And they ended up with not a single case of paralysis in the country. So this was really a huge achievement and a fantastic, kind of the most prominent use of this general concept. So we've been thinking about a number of different ways to use our platform. And we had certainly been thinking along the lines of infectious disease like polio, but we hadn't really settled on just one application. And then SARS-CoV-2 came up earlier this year, and it became really clear to us that we should kind of stop everything we're doing and really target this platform to trying to get the best data we can on the current prevalence and incidence of SARS-CoV-2 in the population, and then we certainly hoped that could lead to some more insight into what sort of interventions we could take. At that time, nobody knew very much about this new virus, so we were really just focused on establishing that causal link. Can we identify the virus in sewage, and does that correlate with the number of new cases? So what I'm showing here, these are data that came much later than the paper I'm talking about. And so this very first paper, this is work done primarily by Fuqing Wu, Amy Xiao, Jianbo Zhang in my lab, but then also a collaboration with folks at Biobot Analytics, so Mariana Matas, Nusha Gailey, Noriko Endo, and also a group that I co-lead with Janelle Thompson in Singapore. So if you look at the graph, you can see that the total viral load that we detect in wastewater actually correlates quite well with the number of infected individuals in that particular catchment area. So some of those are very large treatment plants, like the Deer Island plant that I just showed you. Some of them are quite small plants that serve tens of thousands or even only thousands of people. So there's quite a wide range. But what we found is that the viral load in the wastewater ended up being a pretty good predictor of the number of cases. But when we first looked, at that time, we really thought, after reading the medical literature, that the amount of virus that was seen would be a good measure for how many people are currently sick. And at that point in the outbreak, the number of people who are just infected and the total number of people who are sick, those numbers were pretty similar, because we were really at a very early time in the epidemic, in the pandemic. So we did a calculation. We looked at the total concentration of virus we saw. We looked at the total amount of water that was passing through the pipes. And we couldn't get the numbers to match up with what people were seeing in the clinic. So people had taken urine samples in the clinic, and they basically saw essentially no virus. And then people would take stool samples of COVID-infected patients, and they'd measure how much virus was there. And when we plugged those numbers into the formula that we had to figure out, okay, how many people are really sick, it was telling us that at the very minimum, there were 300 uninfected people for every person who was actually diagnosed. If you want me to guess, 300 is a minimum. I think it was probably more like 3,000 or even 30,000 times as high. And at first, we put out this paper saying that the virus in wastewater is just much, much higher than we would expect from these clinically confirmed cases. And that might mean that we're actually farther along in the pandemic than anyone had realized. So we continued to move forward, and remember, we were kind of breaking new ground. At that point, one other group had identified SARS-CoV-2 in wastewater. I think we were maybe the first group to actually sequence it and get that DNA confirmation that we were really finding SARS-CoV-2. But we didn't really understand kind of that third aspect of being a good wastewater target, right? Like, is the data actionable? And the first step in answering that question is, do we understand what the data means, right? So we decided to continue monitoring the viral loads that we saw in the sewage. And we were able to make kind of the following plot. So what you see here, and this is a great collaboration with all the folks I just mentioned, really again led by Fuqing Wu, Amy Xiao, Jianbo Zhang, and also the team in Singapore under Janelle Thompson. We were able to measure the wastewater virus at the Deer Island treatment plant nearly every day for a few months. And then we compared that data to the clinical cases that were coming up. And what we found is that the virus spiked maybe a week or so before the clinical cases did, and then the virus went back down, and then the clinical cases went down. And when we looked at this plot, what we realized was that we're not really, so we thought we were measuring the number of currently infected people, but when we plot it next to the number of newly infected people, the fit looked really good. And so if we were really at this point looking at how many people are currently affected, what we call the disease prevalence, then it should have gone up and it should have stayed there. And that blue curve should have only gotten higher and higher and higher and then started to level off. But really the blue curve went up and then it went back down, and then the orange curve went up and went down. So the wastewater virus went up and it was followed about a week later by the new clinical cases. And so that started us thinking that maybe what we're looking at is not how many people are currently infected, but maybe when a person gets infected, they shed a ton of virus within a very short window of time, so that when we look in the sewers, we're seeing how many people were newly infected in the past few days. And if that were true, especially back in March when we started to see cases take off, then that could be really valuable, because at that time, we didn't have the testing capacity that we do today, and we weren't really getting as accurate a picture of how many new cases there were over time as we would hope. So we looked into that a little deeper, and what we were able to do is we had a time series showing where all the new infections were happening. We had a time series showing how much virus was in the sewage on any particular day. And so because if we know how many new cases there are, and for each new case we know how much virus they shed on the first day of infection and the second day of infection and the third day of infection and so on, then we should be able to predict how much virus we'd see in the wastewater, right? Unfortunately, we didn't have that middle curve, which is how much virus is each patient shedding each day, on the first day of infection, the second day, the third day. But we had the amount of virus we saw in the wastewater, and we also had how many new cases there were clinically. So then what we could do is we could take those two time series, at least in the Boston area, and we could use some mathematical algorithms to solve an inverse problem, and we could figure out what that shedding function looks like. And that's what you're looking at here. So if you look at the main graph and not the inset, this is how many viral copies a COVID-infected patient is inferred to shed on each day. And you can see on day 0, they're shedding 0 particles because they just became infected. But then by day 1, they're already shedding an enormous number of particles. So over 10 to the 11th. So, almost a million particles. And the shedding lasts for about 3 days. So they shed almost 0 particles. And then for 3 days, they shed something on the order of 10 to the 11th particles. And then on the 4th day, 5th day, 6th day, they're back down to almost 0. Okay. But that almost 0 is actually, those are the numbers that were reported in the medical literature. And so those are the orange points that I'm showing. And in the inset, you can see it on a log graph. On a log scale, you can kind of see those, that orange region with a little more discrimination, right? And so what we realized is that when people get signed up for a clinical study, first, they have to be showing symptoms, especially at that time, when the pandemic just had reached the US. In order to even get tested, you already had to be showing symptoms, and then you had to get a positive test, and then you were enrolled in a clinical study, and then they took a stool sample, and then they measured how much virus is present in the stool sample. Well, by that point, you might be 10 days into the infection, or even longer. And by that time, we're seeing that the amount of virus being shed might be 1 million times smaller than the amount of virus being shed on, day 1, day 2, day 3, day 4. And so this really, even though our previous paper had said, look, the data is indicating that there are many more sick people than is showing up, it turned out that we were actually relying on the wrong information. We were measuring patients after they had already moved on from shedding tons of virus, and we were just looking at the residual shedding. So of course, when we saw these huge viral loads in the sewage, those viral loads were actually just the residual. And so it wasn't that there were 30,000 times as many people who were sick as as had been predicted. It was that each person was shedding much more virus than expected based on these clinical studies that basically enrolled patients too late. So having made that discovery, we realized that we're not looking at disease prevalence. We're not looking at how many active cases there are in a particular region. We're looking at disease incidence, and that is how many people were infected today, or how many people were infected, let's say, in the last 72 hours, right? And so armed with that knowledge, then we could go back and we could start to compare these data with other sources of data that we were getting on the pandemic. So I know this is a lot of information, but if you look at the top track, the blue outline there is, this is how much virus we're seeing in the wastewater every day. The orange track that matches it, that's how many new clinical cases are being reported. In the next track down, we can look at influenza-like illness. So on the left, you see that large peak, and that's reports of influenza-like illness. Or actually the double peak there is influenza-like illness, and the gray peak on the left that almost perfectly coincides with influenza-like illness in the beginning, this is confirmed flu cases. So that first peak we think is that's just the flu, but you can see there's a double peak. There's another peak that comes up around mid-March. And the difference between those is shown in red. And that red curve, the difference between influenza-like illness and actual confirmed cases of influenza, almost perfectly coincides with COVID-19 in the Boston area. So we started to really dive into these data and bring other sorts of data together to see if we could infer, like, what are the interventions, what are the things that are kind of making it better or worse. So we can look at clinical testing, hospitalization, and death. The middle track there with the blue line is mobility. So we can look at cell phone mobility. We can also look at people taking public transit, and that really started to taper off fairly early. Okay, around the time that the governor of our state declared a state of emergency. But we still see the viral titers increasing. We can also look at supermarket visits, and really corresponds with exactly that time. There's a peak when the state of emergency is declared. Everybody descended on the supermarkets, and at that time, it was well before people were wearing masks. So everyone descended on the supermarkets together, maskless. And if we start to look at social media data, we can do that. And we see that there's a lot of postings on, YouTube, Reddit, Facebook, related to health status. And if this is a work done by the SMAP lab at NYU, led by Josh Tucker and Rich Bonneau, they were able to go into those Facebook posts, for example, look at the types of words that people are using and compare those to lexicons of words used by people when they're experiencing anxiety. And so we see all these factors come into play. We see the state of emergency. We see people posting about their health status. We see those people's anxiety is reaching peak levels. They're descending on the supermarkets without masks, and we're continuing to see the viral caseloads surge forward up until maybe mid-April. So that's kind of looking at the region as a whole. And one of the questions that was lingering in our minds was, well, is there an asymptomatic population? Is there any way to figure out whether or not we know there's not 300 or 3,000 times more sick people than have been confirmed, but maybe there's twice as many. Is there any way to actually find out the total number of individuals who are sick. And so we came up with an idea, and that was, the larger a region we looked at, the more likely it is that that region has one sick person. And so what we said is we talked to the city of Boston. We said we want to look at about a dozen different neighborhoods across the city that span a wide range of population sizes. And what we had hoped to do is see that, for these very small populations, there'd be a very small probability of seeing any sick people, seeing a positive case. Whereas in the largest catchments, the largest neighborhoods with the largest populations, we'd be almost certain to see positive cases for COVID, because there were so many people, at least one of them has to be sick, is the logic. So that's what I'm showing you in panel A. And so you can see the different box plots there are arranged by population size, and actually most of the positive cases are in the smallest neighborhoods. And so this was very surprising, and mathematically we checked it out. It made no sense. The data seemed impossible unless there was something else going on. And so then what we did is we looked at the different demographic variables that really describe those different neighborhoods. And what we found in general was that the more socioeconomically advanced or depressed a neighborhood was strongly correlated with whether or not we saw a positive hit for COVID-19 there. And so the strongest predictors of whether a neighborhood had at least one positive case of COVID-19, it wasn't the population size of the neighborhood like we had assumed. It was actually the per capita household income. So higher income, lower probability of COVID. And in particular for wastewater, it was the average number of dollars per year that each household spends on healthcare. So the populations that were spending more per capita on healthcare were the least likely to be impacted by COVID. And so this really kind of hit home for us that, like, this disease, even though at the large scale, so the Deer Island treatment plant has two giant pipes leading into it, right? I said it was 1.3 billion liters of sewage flowing in each day. That comes in when it hits the plant in two different pipes. One services kind of the northern catchments of the Boston area. The other services the southern catchments. And every time the north moved up, the south moved up. Every time the north moved down, the south moved down. And so, and they were moving in lockstep, and they were moving in a way that was completely different than, Miami-Dade County. It was completely different than, New York, New Jersey, California. So, the northern and southern catchments of Boston were truly connected in terms of transmission of disease, and they were disconnected from other geographic regions. But the paradox here is that even within the northern region, or even within the southern region, two neighborhoods that are right next to each other might be completely insulated. So you might have one that's red hot and very high incidence of COVID, right next to, physically next to a neighborhood where there were zero cases and zero virus detected in the wastewater. And so what it said to us is all of these cases are primarily being concentrated in these lower income neighborhoods, probably with much higher density housing and people working in essential jobs that didn't have the luxury of staying home and quarantining. Those neighborhoods that were bearing the burden of disease, whether they be in the north or the south, throughout the entire region, they were in direct physical contact, at least enough contact to facilitate disease transmission. So the disease was sweeping through the Boston region, but in a very particular way, sort of homogeneous at the regional level, but very heterogeneous at the neighborhood level, and it's really the socioeconomics that was determining the spread of disease. So I want to switch gears a little bit now and tell you about some work that we've been doing at Biobot Analytics, and this is a spin out from the lab founded by Mariana Matas and Nusha Gailey. And Biobot Analytics is all about trying to take this public health information that we're gathering from the sewers and make that actionable information and put it in front of decision makers. And so we had been working with the city of Boston and with the region and with the Deer Island treatment plant to monitor virus daily. And so here's another zoom in on the type of data that we're generating. So we're taking multiple samples. In some cases every day, in some cases, running multiple tests every day. And so you can see there's a lot of scatter on any given day. There's a huge range there of values. But one of the things we noticed about mid-June was that even though cases were declining, okay, and this is a logarithmic scale, so a decline here means an exponential decline, and then an increase means an exponential increase. And so we saw this pattern. We saw this sort of declining pattern, and then roughly around June 20th, we saw it turn around, and the slope becomes positive, and the cases are going back up. And this was exactly kind of the early warning that we were hoping for, and to us this was it was very frightening because we live here, but it was also in some sense reassuring that that as long as we kept vigilant and as long as we kept monitoring the virus in wastewater daily, we'd be able to detect these changes. And so the vertical lines here correspond to different phases of the Massachusetts state reopening. And so we were able to get information back to decision makers who could then start to monitor very closely and really start to understand what are the interventions they can take, as part of the reopening, and how are those impacting the number of new cases. And because I said that the virus tended to spike earlier than the new cases came in, this gives us even more of kind of an early warning system on that things might be starting to come back. In the meantime, those data were we were a little unhappy that those data were very messy. And so one of the projects I've been spending most of my time on is a collaboration with Chris Smiley, who's an assistant professor now at Massachusetts General Hospital and Harvard, and also Noriko Endo, who is one of our top engineers at Biobot Analytics. And what we've been doing is we've been taking the algorithms that you might be familiar with from your mobile phone. If you open Google Maps, what you'll see is sometimes you see a big blue dot because GPS isn't that accurate and it doesn't know exactly where you are. And then as you start to, as you leave Google Maps open and, maybe even start to get in your car and drive around, you'll see that blue dot get smaller and smaller and smaller, and you'll see it start to get really, really accurate. As you start to make more measurements and you're combining those measurements together, it's keeping track of your position and your velocity. And every time you ping those satellites and you get some data back, it's going to improve its estimate of where you are. And we said, well, COVID is kind of similar, right? Instead of a position, we're keeping track of how many new cases there are. And instead of a velocity, we're keeping track of, how quickly is the disease spreading or how quickly is it declining? And so in mathematical terms, we can talk about NT, which is the number of new cases as a function of time, and that's what we're showing on the top there, and that's again on a logarithmic scale, and then RT. So most people at this point, if you've been watching the news coverage, you've heard of R naught. R naught is the reproductive number, and it says something about how the virus spreads. But you're also probably familiar with the idea that, the things that we do and the precautions and interventions that we take can actually change how quickly the virus is spread. So if we wear masks, if we obey quarantine rules, if we, limit large gatherings, all of these things are affecting the reproductive number, which you can think of as how many new people does each infected person infect before they, are done with the disease, hopefully with a positive outcome, right? And so if that, so instead of R naught, which is kind of how transmissible it is, let's say at the beginning of the pandemic, we can talk about RT, which is how transmissible is it? What's the reproductive number as a function of time? And that's what's really shown in the bottom panel there. We see that the reproductive number was probably quite high early in March, and then has been pretty close to 1 ever since. Okay. And if you look in the curve in the bottom right, you can kind of see the difference between the number of new cases that are being predicted by our new model and the number of cases that we actually see clinically. So if you look at the yellow dots, those are the earliest. What we see is a lot. The data is telling us in the wastewater that there are many more sick people than are being clinically confirmed, and this occurs throughout, most of the peak up until, maybe mid-April, something like that. And then what you see is it actually goes the other way. And the number of confirmed cases is actually higher than the number that we would predict from the wastewater. And then once we get into the red, which is more recent, the two numbers are pretty close to each other, right? So the wastewater and the clinical data are the same. And this to us was really gratifying, and it made a lot of sense because in the beginning, we didn't have the testing capacity. So we didn't know how many sick people there were because people had a hard time actually getting tested. Some of those people eventually got tested, and those test results came in much later. And that's what we're seeing in the orange region, where the clinical numbers are actually exceeding the wastewater numbers. In these days, we have a pretty good handle on the total number of new cases. And so we see really the same measurements coming out of the wastewater and out of the clinical numbers. So here's another view of kind of the same thing. So on the top in blue, you can see the number of new cases over time in the Boston area. On the bottom in green, you can see our estimates of the reproductive number. But the most important thing here is that dotted line. So that dotted line is a reproductive number of 1. And so it means if there were 300 people infected yesterday, there's going to be 300 people infected today and 300 people infected tomorrow. And if RT is a little bit bigger than 1 or a little bit smaller than 1, then we have 2 possible outcomes, right? And if you look at the forecast there, this is a forecast starting from, roughly, let's say 100 new people being infected per day. If RT and the shading on this graph is kind of our current error bounds around RT. And so if you look at the forecast, we're in this precarious position where the current RT is about 1, but if it's a little bit bigger, then we're going to see this exponential growth. And if it's a little bit smaller, terrific, we're going to see an exponential decay. And right now we're sort of hovering around an RT of 1. And that's kind of now I think folks are sort of up to speed with my understanding of the situation, and maybe we can stop here and just go into the chat and discussion section.
Great. Thanks so much, Eric. That was exciting to kind of see all the progress you've made on that since I last heard about it. And I'll remind folks, we have a few questions in already, but if you will have questions to ask, please enter them in. But I'll kind of use my position to ask the first question myself. It sounds like Biobots in a lot of different cities all over the country at this point are getting data from a lot of different places. And I really was glad to see that the estimates from the wastewater and from the clinical models are kind of converging. And so I'm wondering if you also had cases where there was a community of people where they were almost comprehensively sampled via, say, PCR, and you also had wastewater catchment data for that same population during an outbreak. Are there any cases of that occurring?
I don't know of any cases of that. So it still remains an open question. What's that conversion factor? So I should say, right now we're in a position where we can use the wastewater estimates to understand how many new cases are going to be reported tomorrow or next week, something like that, right? But it might be that for every 2 cases, only 1 is reported. That's something that we don't know, and the type of study that you're talking about, where you get comprehensive data on everyone, like that's the type of information we need. I do think we have some ways to figure that out, and so if you remember, my first idea was looking at populations of different sizes, right? And I thought that was going to be like really clear cut, and we were going to nail it, and we were going to figure out what that ratio is and how many undiagnosed cases there were. But it turned out, to our surprise, that no, it had more to do with socioeconomic status. But I do think that we're still looking into it. And what I would say is the number of folks that are infected today versus tomorrow in most of the cities that we're looking at is very, very similar. So what we can do is we can look at the fluctuation in those numbers. And we know how large those fluctuations should be. If we use something called Poisson statistics, it tells us that if we're looking at 100 new cases per day, then it should be 100 plus or minus the square root of 100. So it should be 100 plus or minus 10, right? And so if we start to see that, no, it's actually 100 plus or minus 20, then that tells us, well, maybe there's less than 100. In this case, it would probably be the opposite. It would be 100 plus or minus 5 because there's underdiagnosed cases, right? So we're still looking into how we can use the size of the fluctuations to give us an idea of how many undiagnosed cases there really are. But it's tricky, and we need to make really precise measurements, and so we're working on it, Sean.
Cool. Well, thank you. Okay, well, let's get to some audience questions. So the first question with the most votes is, how many communities, cities, states are using wastewater epidemiology as part of their guidance for reopening?
Oh, great question. Gosh, I wish Mariana or Nusha were here, because I am spending most of my time on the technical side of things. So I don't actually know that. I know that, here in the Boston area, I know that certainly the state of Massachusetts is taking the data, digesting it, taking it quite seriously. I don't know that it's baked into any specific reopening plans, but I do know the city of Somerville definitely has some of their regulations around school attendance and school reopenings specifically tied to virus titers in the sewage. Nationwide, I have no idea. I know there, at MIT, we're measuring COVID in, I think, 7 or 8 different dorms on campus. And I know at ASU, they've actually identified a positive case in a dorm, then quarantined the dorm and started individually testing the students in it. A lot of businesses are thinking about this, and a lot of different schools are thinking about it as well.
Cool. And so to kind of piggyback off of that, I guess I have a little two-part question that combines another audience question with one of my own. How expensive is this for cities and municipalities to implement? And what kind of information does this give public health officials, right? You said there's like a time lag between when these titers in wastewater increase versus when new cases start to increase. What kind of time can you give these officials to a lag time to start acting?
Yeah, it's a good question. So I want to kind of shift it toward rather than getting the most precise, most detailed information as quickly as possible. That's what we were doing at the beginning of the pandemic. It's like, how can we measure the concentration of virus in wastewater even before we understood what that meant? I think we're reaching a point now where we do understand what it means. And so we're shifting our focus a little, and we're saying, how can we get decision makers information that is actually actionable, right? And that might not be being the first person to observe that the viral titers are getting higher, but rather being the first to kind of have the analytics that can tell us with 99% confidence that the current reproductive number is now greater than 1, and we're going to start to see an exponential increase if we don't take an intervention. So I think it's a little hard. I mean, I can tell you all about sort of the timing of like when we can start to see things, but all these measurements have errors. And so like the real question in my mind is when, how early can you give decision makers information that is reliable enough that they can take action on? And so it's very easy to take sewage at a treatment plant, measure the concentration of virus, and you've got a number. It's maybe 10 to the third viral particles per milliliter of sewage, and yesterday it was half that or something like. But then what do you do with that information, right? And so figuring out when the probability that you need to take action crosses some threshold that then is set by the folks running the reopening or making the important decisions on COVID response. That's kind of that's the hard problem. That's what we're working on now, and we're starting to think about what are the strategies for sampling. So. We may have partners that are sampling once a week, but then if we see an anomaly, maybe we need to ask them to sample 3-4 days in a row, so we can confirm it and get them that information faster. These are the kind of internal discussions we're having right now to help, at least at the company, help those customers get the most out of the data that they're getting back, because this didn't even exist like a year ago. This sort of product of COVID wastewater surveillance. So we're sort of constantly innovating, constantly adding value to the folks who are partnering with us.
Thanks. And kind of bringing it back to, so I'm being a bad host here by not saying the names of the people who are asking questions. So the first question was from Joey. This question that I alluded to a little earlier was on cost, right? So Biobot is providing the service to cities. So Mary Patricia asks, how expensive is this testing for like college campuses or cities, what have you?
That is a good question. I'm more the science guy. I don't get into the business side. But if you go to the Biobot website, somebody it might be on there, or somebody would get back to you with that information.
And this might be a case where I might know a little a bit myself too. I saw a interview by Mariana on the news a while back, and I remember she was saying you guys got some funding, some venture funding, to process a certain set of samples. And so a lot of your partners it was a kind of a pro bono service, but that might have elapsed at this point. I'm not sure.
Yeah, so the pro bono campaign, that was really great, and that allowed me to really collaborate with the company while like through my academic lab, and that ended in, I think, like early June. So up until that point, we were just really figuring out how to use this data. And so everybody just came together. We said, look, we're not trying to make a profit or even like pay people's salaries. And I know they took a lot of their startup funding and put it into just developing this product and getting data back to users. And it made sense because at that point we didn't really even know what the data meant, how to use the data, things like that. And now the company has grown quite a bit in the past few months through offering the COVID product, and so now it's more of a the business decisions get in the way. I don't take part in them, but
oh, Mary can look it up on the website. Okay, so moving on. So Candace from Town Hall asks this question. I can imagine this information being used on the micro level in the home to determine what's missing from a family's diet. Is that a tool or a service that can exist, or something that your lab is looking into as well?
Yeah, that's really interesting. Honestly, I think that would be a completely different. So I think it's a great idea. It's an idea that I've thought about quite a bit. It's an idea that I've even considered starting a different company with Mariana and with other people to do exactly that. I think something like a smart toilet could be a really useful vehicle for enabling and empowering individuals to start collecting and understanding their own personal healthcare data. I think it would always be something that is separate from Biobot, because there's a lot of issues around privacy and what you're going to do with that data. So Biobot is, it sounds like an infrastructure company. I would say it's a data science company. I would say, in the same way that there are companies that, like Google that mine all of the data on social media and other places to derive insights and hopefully improve people's lives. I see Biobot as a company that can mine chemistry data and DNA sequence data, and then use those same types of machine learning approaches to improve people's lives. In order to do that, everything has to be kept anonymized. In the case of Facebook and a lot of these other data science companies, it's never perfectly anonymized, and it leads to problems. In the case of Biobot, the controls that we have in place, even though right now there's not a lot of regulation in this field, so we've tried to kind of keep ahead of the curve by having hard limits on the size of a catchment that we look at. So everyone's data is essentially mixed together and anonymized in a way that is kind of foolproof. Like once it's in the sewer pipe and it's completely mixed together, you can't pull out which part belonged to whom, and that's what we use to try to make those inferences, right? If you start talking about just extending it up and up and up, farther and farther upstream, closer to the toilet bowl, that's what we think of as upstream, right? Then at some point it becomes a privacy concern. And so I think just that makes it difficult. I think those are two totally different offerings. One that is maybe something like a smart toilet that goes inside a user's home and just gives data back to them. And then the other is something that takes this agglomerated data from all these different places, mixed together, and then makes public health inferences.
That makes total sense. Okay, so next question is from Sophie, and she asks a great question, actually. What are some surprising trends you've learned about the spread of this novel coronavirus from sewage surveillance? What are some surprises that fell out of the data you didn't expect to see?
I mean, gosh, it's all been very surprising. Kind of every step along the way. I've tried to highlight what I thought were the biggest surprises, which are, gosh, we went in thinking we were measuring prevalence, like number of active cases. We're actually measuring number of new infections, and maybe some of the surprising things to me were related to some of the other work that we've done, both in the academic project and at Biobot. So we were originally a chemistry company, and so we weren't doing any DNA sequencing work. Mariana and Nusha had this. I kept pushing them. They had a lot of ideas for what that first application might be. And as soon as they came up with monitoring for the opioid epidemic, I was like, gosh, that's brilliant. It's timely. This is the product we should be pushing forward. And just learning more about that has been surprising, kind of every step along the way. So I know that one of the really interesting findings was that if most of the data coming in on the opioid epidemic before apart from sewage surveillance, it's just coming from overdoses and it's coming from deaths. Okay, and so there can be a huge time lag between, let's say, someone becoming addicted to opioids that they were prescribed by a healthcare provider, all the way to many years later when they're buying heroin on the street or fentanyl, they're having an overdose, they're calling 911, and then that data is getting recorded, and that data is getting it back to decision makers, right? So there's a huge time lag, but there's also bias, and so you might be in a really posh neighborhood or something like that, and you're seeing very few like heroin overdoses, and so you're tempted to assume that, oh, it's all all of the heroin use is in the socioeconomically depressed regions, but in fact, oftentimes we'll see high rates of hard drug use in some of these more posh neighborhoods, but then we'll also see high levels of Narcan. And so, it may be the case that people have a bigger support system, and they have a buddy who's keeping an eye on them. And if they overdose, they can deal with it in their own home, and they don't have the shame and the stigma of calling 911. And so, getting better data faster, and getting it in a way that's less biased. It's less biased than having to wait for somebody to contact a healthcare provider. It's less biased than, at the beginning of an infection, relying on confirmed cases when there's no testing capacity. Like to me, it's not even as much surprising as it is, just removing those inherent biases that are sort of focusing us on the wrong thing.
Cool. Thank you. Well, let's see. We may have time for two more questions here. It's 7:11. So the next question, I think, kind of relates to, one of the surprises that I found from your data, which was this bursting sort of observation that you were able to infer from the model. So this Nick Bowman asks, does the amount of viral shedding shortly after infection correlate with disease severity later on? And, you don't have data on individual people, but it is interesting to ask, since you have neighborhood by neighborhood sometimes data, do you see associations between like the bursting rate or these parameters of the model and other information on disease severity from these populations?
Oh, I wish. Yeah, I don't. It's pretty hard to say, and the reason I'm pausing is it's a great question. It's an important question, and I don't want to say it's impossible with the data we have. I think I need to think about it. So thanks, Nick. I won't think about it. But yeah, I don't see a way to get past the fact that it's kind of pre-aggregated to say anything at the population level about what might be happening at the personal level. But I could be wrong. So
it's like atop the top of my head, just thinking about your observations between socioeconomic status and patterns you're finding, and if the model parameters are different for these different populations would be kind of interesting.
Yeah.
Thanks, Nick, for the nerdy question. All right, so I think we'll pass the hour here, so we'll end off with one final question here from Joe Meister. If the prevalence of disease oops, where'd it go? It dropped down. Okay, if the prevalence of disease is associated with socioeconomics, could that lead to different public health approaches within cities and communities? So, do you know of examples of people who have changed their public health policy based on that kind of information?
I don't, and I'm not saying there aren't, but gosh, I mean that's my hope. I think that is that's what we're hoping for when we publish these data. I mean that's really the whole point of monitoring different neighborhoods to take limited resources and apply them where they can kind of do the most good. If there's any decision makers out there, this should be a priority, really. I think we need to think about which populations are bearing the largest burden of disease, and then making sure they have access to the same resources that everyone else does. Great. Especially the folks who are keeping society running while the rest of us are in quarantine. I think those really are the folks who are bearing the largest burden of disease. And I think especially because they got hit very early, that they really haven't benefited from any of the data and any of the scientific advances that have been made, and hopefully we can fix that, and hopefully we can do it better. God forbid the next time that there's a pandemic coming around.
Yeah, and hopefully some of these new monitoring tools allow for the better deployment of these public resources to help these people.
In fact, I think that's where we can add maybe the most value, is figuring out where to deploy limited resources.
Yep. All right. Well, on that note, Eric, it's been an absolute pleasure to speak with you and to hear about all this work that you're doing, and it's exciting to see the progress that's been made. So thanks so much for the great talk and the great conversation.
It's my pleasure, really. Great to see you, John.
Good to see Eric. Okay, so I have a few sign-off things to get through here. Thanks again to our event sponsors, Metabolon and the Washington Research Foundation. And a special thank you to Town Hall Seattle for your incredible partnership in these ISB partnered talks. So I'm going to make a plug on a couple of events that we have coming up next week that focus on the microbiome. And this is maybe not for a public audience so much, but for scientists that are in the audience. On October 15th and 16th, we'll be hosting a microbiome data analysis course and symposium on the human microbiome and its role in precision medicine. Both events will be free and virtual. And you can learn more about them at our website, which is, again, ISBscience, all one word, .org. microbiome 2020. So if you want to participate in those events, I encourage you to go look us up. Also, please mark your calendars for the next ISB Town Hall Speaker Series. Thursday, October 20th at 6 p.m., we will hear from brain scientist and best-selling author Dr. David Eagleman on the topic of brain health. We hope to see you there. Please learn more about ISB at our website, like I said, isbscience, all one word, .org. Thanks to everyone for joining us tonight, and have a great rest of your evening. Bye-bye.